Well, while people are rolling in, let's get started and do some introductions, and then we'll set the stage for the rest of our presentation. So thank you, everyone, for joining us. It's a pleasure to be with you. My name is Brennan Duty. I'm a Director of Business Development at Ginkgo Bioworks in our agricultural practice. And I came to Ginkgo after sort of up and down the value chain within agriculture. So stints at Cargill, Land O'Lakes, Winfield. I also had a startup that was focusing on an enzymatic approach to controlling pests in agriculture. And before doing my MBA at Cornell, I lived and worked in Latin America, especially Brazil, for several years, which, of course, for many on the call will realize it's the huge market for biologicals. That's a little bit about me. Magali, I'll kick it over to you, please.
Hello, everyone. Thank you so much for joining us. So my name is Magali Guilabert, and I am the head of the Ag Biological division here at Ginkgo. I've been with Ginkgo for a year. I came from the acquisition of the West Sacramento site from Bayer Crop Science. So I worked for Bayer Crop Science before for 12 years. And before that, I was working for AgraQuest, which was a startup company that got acquired by Bayer. So I have always worked in agriculture. I have always worked in biologicals. I've worked obviously for a small company, bigger company. So I truly understand what it means for a small company or a big company to have a dynamic portfolio and a very nice pipeline. And before all of that, I was doing my PhD at UC Davis in plant pathology. And I specialize in microbiogenetics and plant microbe interactions. And before that, I was studying in France, plant breeding specifically. So I have a well-rounded, solid knowledge around ag.
Thank you, Magali. Andy.
So hi, I'm Andy. I'm a senior foundry lead at Ginkgo Bioworks. I'm leading the encapsulation and screening foundry, and I'm happy that I can tell you today a little bit more about the technology platform that we are using. I joined Ginkgo like 1.5 years ago through the acquisition of FGen. I was before heading the overall activities of FGen since about 2010, so 10 years. And FGen really used high throughput screening for strain development and protein engineering and protein development in several verticals, from food to industrial biotech, but also to agricultural biotech. And I'm happy to be here and tell you a little bit more about the technology today.
Thanks, Andy. Thanks, Magali. I'm really happy to be here with you here today and appreciate that. So on the agenda today, we're going to touch on a few things. The first one is the challenges that growers are facing today in terms of the innovations that need to happen in order to keep abreast with climate change and how innovators like yourselves are working with companies like Ginkgo to address those challenges. Then we'll talk about Ginkgo at large, and then we'll also talk about Ginkgo in terms of an agricultural company that's providing R&D services and how our platform can help your efforts towards commercializing better biologicals. Then we'll also dive after that into the unique technology which really brings us here today. And that is, how can we use this technology to rapidly improve biologicals performance and get a second generation product into the hands of farmers that's more efficacious and can help them win in the field. Then we'll close with an overview of how we partner at Ginkgo in terms of the commercial structure, as well as intellectual property considerations. And Magali and Andy will walk us through some case studies in terms of how this technology has been applied in areas adjacent to agriculture, but nonetheless applicable to agriculture, and Magali will discuss that specifically. So let's get started. So as everyone on this call is aware, growers are under a lot of pressure. Everything from trying to grow more food on the same amount of land, dealing with an erratic climate, changing regulatory regime. There's a lot that growers have to address. And it's the charge within us here at Ginkgo, as well as our innovation partners, on innovating our way to solutions to meet the demands of growers for them to be able to meet these challenges. And so many of you are familiar with or are actively developing biological solutions to address these challenges. And biologicals for Ginkgo really means live microbes or products that can be made by microbes, such as proteins, RNA, pheromones, plant growth regulators. And they've long been part of the solution set to these challenges. And these products have huge potential, but growers expect biologicals that are potent, consistent, and stable on the shelf. In other words, they want them to approximate as best they possibly can to conventional chemical inputs. And so what we believe at Ginkgo is that we can take the tools of biology, synthetic biology, and really take these things from what have traditionally been conceived of as worse cost in use and less efficacious, and boost their performance such they can compete with or complement conventional chemistry, giving farmers more choices and enabling them to win on farm. Of course, the path to developing a high-performing, consistent biological product is rife with technical risk and subject to a lot of oversight. So our aim is to minimize those risks in the innovation process, de-risking these projects really quickly and delivering products that meet the expectations of growers and regulators alike. So today we're going to focus on our proprietary technology to dramatically improve the performance of biologicals quickly. So quick, in fact, that you can have a product in hand to test by the next growing season, all without rational genetic engineering. In other words, it's genetically modified friendly for regulatory regimes. That could either be a headache or a showstopper for regulatory, depending upon your markets. So before we dive deeper into our rapid strain optimization and really even who is Ginkgo and the agriculture perspective, let's take a high-level view about who is Ginkgo as a company. Ginkgo started in 2008 with the premise of, what if we concentrated all of the world's biology onto one platform so that we could learn from pharmacology, nutrition, wellness, and agriculture, and have these learnings go across these different projects in the service of doing projects ever faster? Because after all, pharmacology, which is medicine for humans, is very applicable to crop protection, which is medicine for plants. So bringing all of these ideas and R&D projects onto one platform would allow Ginkgo to be able to utilize these learnings across projects and do projects faster, better, and de-risk them faster than you can necessarily by yourself. That's the premise about Ginkgo by putting this onto an R&D platform. Now, you might have been reading about some of the acquisitions that we've made in the team that we've been building within agriculture. And indeed, that's what brings us to the ag side today. So the ag part for Ginkgo is where we support product development from discovery all the way through scale up. So we support innovators through the product development cycle, from better starting points for new products to services, to services that improve and optimize product performance, and services that deliver more consistent and stable products. So we start that on the far left-hand side for two different areas. The first one is a proprietary strain collection at 220,000 isolates strong. And what innovators can do with this is have us search these isolates for novel compounds to target certain pests. And we can also use our metagenomic database that spans more than 5 billion genes, 95% of which are not available in publicly available libraries, and 99% are sort of unknown to science that are characterized from microbes, to be able to find countless protein and pathway designs for metabolites that can show promise in terms of attacking pests, as well as other novel chemical compounds. This data enables us to approach strain engineering intelligently. We can then curate the protein and pathway designs that we think are most likely to deliver the characteristics that you need from your biological products, and then test them in a highly automated lab. These approaches for innovators who wish to employ cisgenic or transgenic engineering and supercharge their biological products. And when we think about transgenic engineering within Ginkgo, especially within agriculture, of course, you could deliver a transgenic live microbe to the field to do, let's say, nitrogen fixation, but we're also talking about putting these pathways into host organisms to produce metabolites, chemicals, on demand. So our discovery and optimization services are really just half of what we offer. We want to help you mitigate the risk of your products failing in field trials or losing their potency in manufacturing or storage. To that end, our formulation and fermentation process team sits on the same campus as our in-plant testing facility in West Sacramento. This allows for iterative processing and testing that we can do across a variety of product concepts and test the performance in growth chambers, greenhouses, and microplots, so that by the time that we deliver to you a product to do field testing, you know that it's been quite sufficiently de-risked all the way from the lab to the field and the microplot. So broadly speaking, our aim as your partner is to help you get higher performing, consistent products to the market faster. So now that we've given you an overview of all of our platform supports, we're going to zoom in on how we optimize strains rapidly. Specifically, we want to tell you about our encapsulation and screening technology, aka NCAPs. Paired with Mutagenesis, a well-known technology, NCAPs can help you identify the strain variants that can have up to 5x performance and improvement. On that note, before we get started into that area, I wanted to share with you some really delightful news that we just signed a deal to work with Agravali. It is a leader in ag biologicals in Brazil, and they signed a multi-year collaboration to develop next generation ag biologicals with us, including what's using the service that we're talking about today. So as part of this collaboration, Ginkgo will use NCAPS technology to optimize some of Agravali's products, and we're super excited to work with these vanguards of innovation and manufacturing prowess in Brazil. And if you're listening to the call today, thank you very much for your partnership with Ginkgo. So now I'm going to pass it over to Andy, who's going to introduce the technology and how we pair it with massive strain libraries to drive rapid strain optimization. And a quick note about some of the case studies and the examples that he's going to give is that if you recall when I said that Ginkgo works across all these different areas, agriculture, nutrition, pharmacology, and other areas, we have worked in agriculture, but the case studies that we're going to be talking about today are going to be from different areas which we can publicly talk about. So the agricultural case studies that we could elucidate later, more than happy to invite you to a private conversation and we can discuss that more in person. But these are the case studies that have relevancy to agriculture that we'd like to go through today. So enough for me, and on to you, Andy.
Hey, thank you, Bren. So let's talk about NCAPS. What is it and how does it work? So very broadly spoken, NCAPS is a screening technology that can screen millions of strains at once. Those of you who are familiar with biological R&D might be thinking, well, sure, I can use a fax to screen all of those variants in high throughput. But let's say that your biological product is a live microbe that secretes some active ingredient, or maybe your biological product is an insecticidal protein and you need a strain that hyperproduces the protein to bring your cost of goods down. So fax alone will not work to characterize rates of secretion. So with NCAPS, we can encapsulate each strain variant in its own nanoliter reactor. So on the slide here on the right-hand side, you see such nanoliter reactors, which are typically 200 to 500 microns in size. We link the desired phenotype to a fluorescent signal depending on the activity of interest that we are looking for. Increased activity may correspond to a gain or a loss of the fluorescence. So in this picture, you see with a red stain, you see microcolonies of microorganism grown, and the green signal will be the readout for the good strains. So inside the nanoliter reactor, the cell can proliferate and secrete its products, be that a small molecule, an enzyme, or a protein. We pass the nanoliter reactor through a flow cytometer, and those with the greatest change in fluorescence signal are sorted out. So the strains is released from the nanoliter reactor after the sorting, and the activity can be validated in a secondary assay, just as in a suspension culture. So what's so special about this? There's a few things I think is really worth highlighting. So we are in an area where we have affordable and fast ways to generate a lot of genetic diversity from a starting strain. So classical mutagenesis is a commonly known technique among agricultural innovators. But even if you want to take a cisgenic or transgenic approach, you can very easily design and build very large combinatorial libraries. So the bottleneck, if you have such large libraries, is typically the analysis of these libraries, so the screening. So with NCAPS, there is some upfront work to design an assay that really fits to the phenotype that you are looking for. But once that is complete, the mutagenesis and the screening happens very quickly because we can use tried and true biological methods and equipment. So we can typically deliver strains with dramatically improved performance validated through in vitro screening in a time frame of approximately 7 to 9 months. The other reason why this technology is really special is that it's compatible with a broad range of microbes. So you can use any favorite bug that you have in your lab. It works with bacteria, yeast, fungi. You can try insect cells. It's really, there is no limit in what kind of cells we use in the platform for the screening. So NCAPS has the unique ability to screen individual cells that are secreting a product at ultra high throughput. So we can screen for various phenotypes that are related to secretion. In another assay, we can also look for intracellular products, but I really want to highlight here that we can screen for the secreted products because they are more relevant to the ag biosystem. For example, we can screen nanobody variants for their binding activity, we can microbes for their antifungal activities, or strains for nitrogen fixation or phosphate solubilization. So after the introduction of the technology, I'm going to walk to two case studies that demonstrate how this technology really can deliver value. Magali will then walk through how this prior work is applicable to product development in agricultural biologicals. So the first case study is from a partner in the food industry. So our partner was producing an enzyme through a fermentation process with a proprietary fungal strain. So to achieve a better cost of goods, they wanted to optimize the strain's enzyme production. And to have a quick path to market for an improved strain, they were only considering optimization method that leveraged mutagenesis or other random approaches, so-called the classical strain improvement way. So our first step was to onboard their strain and design an assay that measured improved enzyme production through fluorescence. Since we always want to directly measure the targeted phenotype, we use the fluorescently labeled protein for determination of protein activity. So you can see here in the middle microscopy picture, we used a red fluorescently labeled protein substrate. And you can see the protease activity where you have a reduction in the red fluorescence. When we combine these now with the biomass staining, which you can see on the left picture, we can calculate the specific activity and really sort out the strains that produce most of the protein per biomass. So degradation of the protein results in the loss of fluorescence, which can be used then as a metric to determine protease production levels. So from there, after the assay was developed, we generated a library of nearly 1 million variants from their starting strain using classical UV mutagenesis. We then encapsulated each variant in nanoliter reactors with their appropriate growth and assay condition and sorted out the variants that demonstrated the highest level of enzyme production based on the assay principle. So among the strains that we screened, we isolated the top hits and performed the characterization, as explained before, in a liquid culture with respect to protease production, and which confirmed that we isolated strains with improved product titer. So we delivered some of the strains to our partners. They tested 3 in pilot scale, and then based on different parameters, they selected 1 strain for industrial scale-up, and this strain that they're currently using in the production process has about a 30% improved titer and thus a significantly reduced COGS for the product. So Magali, I know, and as Brennan said, this is not a real agricultural example, but does this case study bring up any ideas of how such a technology could be applied to ag biologicals?
Yes, thank you so much, Andy. Yeah, absolutely. So there's a couple of things that actually jump out of me from this program that could be applicable or are applicable to ag biologicals. So for example, many biological products start as a Y-type strain. Usually they are isolated from the environment. And these strains naturally secrete something like a natural product that is going to protect the plants or provide some benefits to the plant as it is growing. So now innovators in the space usually are very familiar with the need to increase performance of those Y-type strains, and therefore to use mutagenesis to do so as a tool to increase the performance. So it's a very fast way to generate a lot of genetic mutation without spending a lot of time to develop tools to engineer those microbes, which usually are going to be extremely difficult to transform. So as you mentioned earlier, once you have a massive library, then you are faced with the problem of how do we test this whole library. And as I was mentioning before, I worked in this field for many, many years, for over 15 years now, and that's always been a problem. So when I came to Ginkgo, I was actually extremely fascinated with this specific technology, because it's truly the solutions to be able to test very, very rapidly a huge generation of different variants. We have to find perhaps 5 or 10 highly performing strains in a million mutant library. So that's really truly to find like a needle in a haystack. And when you have shown with this study, the encapsulation technology truly answer that questions. So it really enables innovators to be able to find and to test every single mutation possible, and to truly find the outlier variant that's going to outperform the others. And most importantly, is going to be in a very commercially relevant timeline. All of that is possible to do, of course, but it will take a lot of time, and encapsulation truly, truly accelerates finding that.
Hey, thanks a lot. So the second case study used a different assay principle. So we call this actually a co-encapsulation assay. So in this case, we were looking for novel antipeptide with changed antimicrobial properties. As humans and animals face pathogens that are increasingly resistant to traditional antibiotics, new antimicrobial compounds with high potency are desperately needed. So in this case study, we wanted to test a large peptide library for its ability to kill pathogenic bacteria. So we thought the most direct way to test this would be to co-encapsulate the peptide-secreting host cell with a representative pathogen model in a nanoliter reactor. So you can see the assay principle here on the right-hand slide, on the right-hand side of the slide, where in blue there is the candidate cell, and in green, there is the so-called biosensor cells. And the principle is that if the biosensor cell is inhibited by the growth, inhibited in growth by compounds that are secreted by the candidate, it will lead to a lower fluorescence. Okay. So essentially, a nanoliter reactor that were the least green contained the candidate cells secreting a peptide that was most effective at killing the pathogen. So we can also see these here on the microscopic picture. In red, you see the library clones, and in green, you see either the inhibition of the growth of the biosensor strain or not. So we ran this co-encapsulation assay and found in the library 126 hits, which forms the left peak of the graph on the right-hand side. So to test the rest of the variants, which on the right-hand side of this so-called histogram were peptides that were not very effective at killing pathogenic bacteria, or that were not produced by the candidate at all. So from the 126 top-performing peptides, we tested them further with respect to peptide secretion levels and minimum inhibitory concentration for growth inhibition of a series of pathogens. So these experiments showed and highlight that we could find efficiently modified novel molecules with improved potencies against several strains. So Magali, I think here the connection to the agriculture is a little bit more close. So might this be relevant for innovators in ag biologicals? What do you think?
Thank you, Andy. Yes, absolutely. So as we know, crop protection chemistry is facing similar problems to the one that you just described. Pests are becoming resistant, for example, to chemical input. So therefore, we see a huge opportunity to develop more biological products to enter the market and to address that specific issue. And we definitively want to be able to help innovators to use such technologies as biological products. So I think what is very cool about the case study that you just described is it is definitively very applicable to develop biological fungicides. And I think here what is truly exciting to me is you can co-encapsulate a variety of different pathogens with the specific small molecule or antipeptide in the specific case, which truly shows that how broadly applicable this tool can be. You do not need to know actually the actual, or you don't need to have an actual small molecule or active ingredient. You can actually use different type of cell lines. And you can use the same principle, for example, to co-encapsulate the variant of the specific starter strain that you have with the fungal pathogens or any other type of pathogens that you want to improve your specific starter strain against. And we can use that. And then the pathogen would be used in this case as a fluorescent biosensors. So what we will do is we will then screen for and isolate the strain variants that have increased potency against the fungal pathogen or any other pathogen that you are looking at. And then, of course, those variants will be generated through mutagenesis, as we talked about before. But we don't have to use just mutagenesis. We can also generate computational libraries, for example, where we can generate a library of amino acid sequences or different type of sequences if the active ingredient is a protein. There's a lot of different ways that we can apply this co-encapsulation technique in service of ag biological innovations. More broadly, we can encapsulate cells with other types of biosensors to test for potency. It doesn't have to be whole microbial cells. It could be insect line cells, for example, or any type of cells that the innovators need. And instead of validating, use the MIC assays, which can take a lot of time. We can also use some of other high throughput assays to validate the hits from these nanoliter reactor screens. So this is truly a very, very powerful technology to develop very quickly commercially relevant products.
Thanks so much, Magali. I really appreciate that. So when we're talking about encapsulation technology and how it applies to ag biologicals, that was super helpful. So let me sketch out what this looks like to work with us. So phase one, which is really months 1 through 4, is really about us onboarding your strain and the cultivation conditions for our Encaps team. So we budgeted about 4 months of work for that. Once that assay is defined, we then mutagenize your strain and develop a strain library. We encapsulate each of those variants in a nanoliter reactor, and with its assay condition, allow the cells to proliferate in these reactors and then conduct the screening. We then take those top hits from that first round of screening and do that same process again. So our experts recommend 2 rounds of mutagenesis to continually boost production and efficacy and really achieve that dramatic improvement that you're looking for that can translate to higher performing products in the field, lower cost in use, lower use rates. Those top hits that are emerging from the second round of mutagenesis are then screened and validated in additional assays. And we can even take that for months 4 to 5 after that in phase 3 to do in-planted validation to confirm that there wasn't any adverse effects in your fermentation protocol, your formulation protocol, and indeed show some efficacy or improved efficacy in planta. So we've described sort of the technical work and how we can partner with you to do that, but you're probably wondering about how much this costs, right? So at the very top of the hour, I mentioned my startup background. And the reason why that's relevant to this conversation is because I know what it takes to work across all different types of organizations from a cash flow perspective, deal structure, et cetera. That's what I work on a daily basis. So absolutely more than happy to work with you. For these types of services that we're talking about today, sort of a starting price is around $500,000 to get you a generation 2 product in potentially less than a year that doesn't have as many regulatory constraints as would a rationally engineered strain and has significantly boosted in performance. So that's what we're looking at from a cost perspective and an intellectual property perspective. Given that this is your starting strain, background is background. That means it's yours, of course. And the improvements upon that, I think you'll see that the intellectual property regime makes a lot of sense. So thank you very much for spending a half hour with us today. We really appreciate your time. And please reach out to me with any questions or to get started about a conversation on how we can optimize your strain and turn it into a second generation product with dramatically boosted performance. Thanks very, very much to Magali and to Andy for joining me. I look forward to hearing from you all soon. Thank you.